Hello, everyone. Welcome to LinkedIn San Francisco. It's great to have you. My name is Farhan Sayed. I am the Vice President of North America Sales for LinkedIn's Learning Solutions business, and it is a pleasure to be here. I'm personally excited to have the Grad Slam here and have the pleasure of introducing our MC in just a moment. There's two reasons why I'm excited. First of all, I'm a graduate of UC Berkeley myself. And yes, go Bears. That's right. I majored in molecular and cell biology, and actually, Alan Sachs here was one of my professors. This would have been awkward. I did do well in his class. He doesn't remember. It would have been awkward if I didn't, so that's a close one. But the reason that I'm excited from that perspective is because I did major in a science, and yet now I transitioned to the business world. And so it's intriguing to me to see what's happening here today with that underpinning of this idea that you're taking research in a variety of fields and thinking about real-world applications and making it simple for people to understand. And so I appreciate that personally due to my own journey from one field to another. The second reason I'm interested and excited for this event to be here is because of LinkedIn's stated vision. Our stated vision is to create economic opportunity for the global workforce. So what does that mean? That means that we're providing a platform for our members, of which there are over 600 million, and ideas to start to meet and connect with each other to realize that stated vision. And what it also means is that what's happening here today with the Grad Slam is very much in line with what we do as a company in a day in and day out basis, which is why we always love hosting this every year. If you will allow us the pleasure of doing that in perpetuity, we would love that. Today we'll be learning a lot about groundbreaking research distilled into bite-sized 3-minute pitches. I'm very excited to see how that goes. And the thing that's important for me, coming from the business world, is to see and appreciate the skills of our students to be able to do that, to communicate that. I know there's a lot of hard work that has gone behind this research, but how do you communicate it in an easy way for people in the quote-unquote real world to understand? That's an important skill. And without that skill, some of this research may never leave the lab in the first place. Through programs like the Grad Slam, this research is being shared, understood, and acted upon, and that's important. But thinking even bigger, perhaps some of these ideas will eventually become a reality and employ hundreds or thousands of people out there. And that's creating economic opportunity, as we talk about in our vision as LinkedIn. And that's inspiring to me, that you could be doing that for the benefit of other people, not just employing people, but also the benefit of your ideas for all of humanity. So we value the work you've put into this research. We value the efforts you've put in to distill it down to these 3-minute pitches, but we also value the potential impact this research has on our global community. And we wish you all the success today, and may the best pitch win. And now I'd like to introduce President Janet Napolitano. Janet Napolitano became the 20th president of the University of California in 2013, the first woman to serve in this role. As president of the University of California, she leads a system with 10 campuses, 5 medical centers, 3 affiliated national laboratories, and a statewide division of agriculture and natural resources. She is a distinguished public servant and is the former Secretary of Homeland Security and 2-time governor of the state of Arizona. Please join me in welcoming President Janet Napolitano.
Welcome to Grad Slam. Yeah. Thank you, Farhan, and thank you to all of you for being here today. This is one of my favorite events during the year because we get to hear today from some of the university's brightest minds, and I'm thrilled to be able to MC us as we go through the program. I want to thank our gracious host, LinkedIn, for once again providing space and support for Grad Slam and for their partnership with the University of California to make this event a big success. You know, Grad Slam offers a wonderful opportunity to pause for a moment and just celebrate the purity of discovery, the pursuit of truth. In the course of the ordinary day, we don't often think about the blood, sweat, and tears that go into the research that affects our lives, from the internet to new types of citrus fruit to our ever-evolving understanding of the human psyche or human history. We have graduate students to thank for many of these discoveries and innovations. And as one of the world's great research institutions, the best public research university on the planet, the University of California is in the business of creating new knowledge and discovering research solutions for big global challenges. You know, as president of the University of California, I am passionate about making basic research more understandable, accessible, and exciting to the world beyond the classroom or the laboratory. I think that's especially important today because, as many of you know, the future of federal research funding remains uncertain. We need to continue to urge policymakers in Washington, D.C. to invest in basic research because it is key to our nation's scientific and technological advancements and to the health of our economy. And we need to continue to show the public the importance of this work to their everyday lives. The 10 graduate students we will hear from today are already champions of explaining their research to the public in engaging, dynamic, and understandable ways. They have proven that they know how to explain complicated research to those who do not conduct research. And they're well on their way to becoming research ambassadors, something our country sorely needs. I'm so proud that these students are part of the UC family, and I'm excited to hear about their world-changing ideas today. Now, before we meet our 5 distinguished judges, I'd like to take a moment to thank Thermo Fisher Scientific for being here today. They have generously sponsored all of today's Grad Slam cash awards, so thank you. Now let's meet our panel of judges. I'm going to ask them each to take a minute to introduce themselves. You can read more about their backgrounds in the Grad Slam program. Jessica, why don't you begin? Stand and face the audience.
Thank you, President Napolitano. Hello, everyone. My name is Jessica Aguirre. I'm from NBC Bay Area News. I anchor the 6 and 11 o'clock news. I am the veteran on this panel today because I've done Grad Slam before, and it is a treat. You are going to enjoy it so much. It's just such a pleasure to be here, to be able to witness the type of innovation that the UC system in California is doing. California has got this right when it comes to schools, that is for sure. And I'm delighted to be here because I know by osmosis, by the end of today, I'm so much smarter than when I walked in.
Good morning, everybody. My name is Alan Sachs. I'm the chief scientist at Thermo Fisher Scientific. It really is a pleasure to sponsor this. In the private sector, we hear pitches all the time. Today, I'm going to very much enjoy hearing pitches about science, which I can fully understand. So thank you for the invitation.
Hello, everyone. My name is Deborah Cullinan, and I am the CEO of Yerba Buena Center for the Arts, which is just a couple of blocks away. I invite you to come over when you're done here. YBCA is San Francisco's Center for Art and Civic and Public Life, and we believe that everything starts with curiosity, imagination, and inspiration. So I am super excited to be here, and like Jessica, I expect to leave much smarter.
First, I just want to say it's a pleasure to be here in this room. My name is Serenity Bassett. I am from Sacramento, California, and I'm here proudly representing Black Girls Code. Yes. Thank you so much. I am a senior graduating high school at Intercom High School in Sacramento, California. I'm also a part of the International Baccalaureate Program and National Honor Society. And thanks again. It's also a pleasure to be here.
I'm John Perez. I'm a member of the Board of Regents. Happy to be here. And one thing Serenity forgot to mention was she'll be starting at UC Merced in the fall.
Thank you, judges. We also have some of Serenity's colleagues from Black Girls Code with us today. Give them a hand. And also with us today are some talented undergraduates that we hope we will one day see as UC graduate students and competing in Grad Slam themselves. So please welcome undergraduate scholars from Berkeley, from Davis, from Irvine, Riverside, San Diego, Santa Cruz, and San Francisco State. Welcome. And there is one more special judge. That is all of you. Whether you are with us here at LinkedIn or watching our live stream online, our audience will have an opportunity today to participate in Grad Slam. Once again, we will award a People's Choice Award, along with the winners selected by our judges panel. After the presentations have concluded, we'll provide instructions on how you and our audience and those watching online can vote. So keep track of your favorites and help choose the People's Choice Award winner this year. Now, the research these graduate students are about to present to you represents years of hard work. So let's give all of our contestants a warm welcome this morning. And now, I'd like to welcome our first contestant to the stage. Give it up for George Deegan from UC Santa Barbara.
Have you ever successfully put a Band-Aid on when your skin is wet? It is really difficult to get things to stick underwater. And the reason for that is that water fundamentally changes the way that adhesives interact with surfaces. So for example, materials that normally stick well in dry conditions, such as Scotch tape or geckos, don't work very well when they're submerged. And this can be an issue because the majority of the human body is water and surfaces in the body are wet. So when you're injured and you need surgery, a traditional adhesive normally won't work. And instead the doctor has to use stitches or sutures to repair your tissue. And stitches are normally not a problem until you need stitches on delicate tissue, such as in fetal surgery, where stitches can do more harm than good. So it would be advantageous to replace stitches with an adhesive that works in wet conditions. And fortunately, nature has already solved the problem. The ocean is wet and has an acidity and salt concentration similar to the human body. And despite this, marine mussels are able to stick to almost any surface underwater. To do this, when a mussel finds a surface, say a rock, it extends an appendage called the mussel foot. And this foot searches around on the surface of the rock, and even now, no one knows what the foot is searching for. But when it finds a spot it likes, it puts the foot down, and it injection molds a liquid mixture of proteins and other molecules onto the surface. And then the foot withdraws. And as the seawater rushes in, the whole thing solidifies, forming an adhesive plaque connected to the rock, attached to the body of the mussel by a thread. And this plaque thread architecture is amazing because the plaque perfectly channels force into the thread, and the thread acts as a shock absorber to dissipate energy and prevent the mussel from being torn from the rock by the waves. This allows it to live in the turbulent intertidal region between land and ocean. So in my research, I studied the interface between the plaque and the rock, and specifically the proteins that localize there. And I asked the question, how are these proteins able to stick in wet conditions when a Band-Aid can't? And I hope that by answering this question, I can guide the design of future adhesives for use in the human body to replace stitches. Thank you.
All right, now we do our spontaneous photo. All right. So your presentation had a lot of muscle to it.
Well, thank you.
Yeah. How did you get interested in doing this?
Oh, well, so I grew up in Wisconsin. I went to school in Pittsburgh, and then I came to California, and I was really excited about that because UCSB is a great place to do marine research and muscle-inspired research. So I'm in chemical engineering, but I work with people in marine bio and in chemistry and the materials department, all working on studying this system, the marine muscle. And there are so many things you can learn from it, and so UCSB is the perfect place to do that.
So you grew up in Wisconsin and Pittsburgh?
Yeah.
How did you get interested in marine stuff?
Oh, well, so actually it's, so muscle research, if you can learn how they stick, you can copy that, but you can also prevent them from sticking, which is it's relevant to the Midwest as well. Because actually my grandmother knows I study muscles, and so she sent me an email where she was like, George, I just read in the newspaper that they're trying to figure out a way to prevent zebra mussels from sticking to things. So it's a freshwater mussel in the Midwest.
Yeah,
and,
it's an invasive species.
Yeah, it's invasive. They spread through lake and river systems, and they clog intake valves in dams, and they cause dam fires. And so
the mussels are on fire.
No, so the mussels prevent the cooling system from keeping the dam cool, and then the dams catch fire somewhere in there.
Oh, that's bad.
And it's bad.
Yeah.
So I had heard about zebra mussels and that kind of problem, but then this is just studying them in a little bit of a different context for medical adhesives.
So what, George, what do you like to do when you're not studying mussels?
Oh, well, I like to run. So I run a lot. I was on the cross-country and track teams in undergrad, and I bake bread too, sourdough bread.
Really?
Yeah.
So here it comes. So I hope you win a lot of dough today. Yeah, okay. This is a tough crowd. Okay. George, do you have brothers and sisters?
Yeah, 3 younger.
3 younger? And are they also interested in science? Or how did you get interested in science to begin with?
Well, actually, so my siblings, my little sister, so the younger of the 2 sisters, she is starting at Northwestern for graduate school in systems bio in the fall, so I'm really excited for that. And then interested in science, I liked math and chemistry in high school, and then I kind of just continued following it. And chemical engineering is very flexible, so at any point I was like, okay, I could go into industry or research and study a lot of different things.
Yeah.
But I'm happy I ended up here.
We're glad you're here too. And what are you interested in doing after grad school?
Well, I want to do a postdoc and then go on the faculty search.
There you go. Where you can also make some dough. Yeah, it just happens. Anyway, congratulations.
Oh, thank you.
Good luck.
Thanks.
Okay, now I'd like us to welcome our second contestant to the stage, Angela Nicholson from UC San Diego.
Inside each one of our cells, millions of particles are constantly bustling with activity. All of this activity is organized by a molecule called RNA, shown here by these yellow dots. Each piece of RNA is an individual written message that your genome uses to transmit information out into the working cell. This message system directs everything from production of muscle fibers to protecting you from viruses. When your cell first makes this RNA, it synthesizes just the main message portion. After that, a tail gets added to each RNA at a predetermined long length. Traditionally, it's been thought that the tail protected the main message, so long was viewed as better. And it got written down in a textbook in this way. RNAs have long tails. And sometimes, once something is written down in a textbook, it's just assumed that that's the end of the story, and it doesn't get questioned anymore. But sometimes, you find something totally unexpected in your research. Our lab has recently discovered that while RNAs might start out their life like this, they wind up with a myriad of tail sizes, and actually short tails are the most common. But why would a cell bother to make a tail long to begin with just to shorten it up later on? By following these short-tailed RNAs, I've discovered that they actually play a very specific role in signaling to the cell that this particular message should be communicated quickly and effectively. By allowing for long tails on some RNAs and short tails on others, tail length can now be a key part of differentiating each message, being specific to what the cell needs. This finding turns the textbook dogma on its head. Now, I'm talking about a very small molecule, and even smaller changes in this tail region, but these findings implicate tail length as a vital part of the message. For an example of how important this is, I want to talk about Fragile X syndrome, which is a major cause of intellectual disability. Recently, it's been discovered that patients with Fragile X have RNAs with different tail sizes than those who are not affected. As you'll remember, the prevailing view has been tails are long and have no effect on how each message is communicated. So how could they be involved in a disease? Based on my work, we now know that tail length can have drastic effects on development. So working to understand how tails are misregulated in Fragile X could lead to some really important new treatments. This seemingly tiny change on a tiny molecule can affect how the entire human body is functioning. This is just one example of how my research could give us a new lens to reevaluate some very complicated diseases. On top of the scientific implications, this work speaks to the importance of reevaluating long-held beliefs in any field. Scientists once thought that the tail of an RNA was just a passive bystander in regards to how each message was communicated. Turns out, it's more a case of the tail wagging the dog. Staying open and curious in our research could lead to some really big breakthroughs in the coming decade. Thank you.
Congratulations. Okay. All right, so what got you interested in researching RNA tails?
Perhaps the most exciting thing about it to me is that RNA and cell biology is kind of at the base of all biology. So no matter what area you work in science, often this will relate to some area of that because it's such a fundamental aspect of what every single cell is doing. So I think that's really exciting about it, that it affects all areas of biology.
Yeah, so have you always been interested in science? Like, did your family talk about RNA at the dinner table?
No, I don't know if they could even tell you exactly what RNA is still. None of my family is directly in science. I actually didn't get interested in science until I was at community college, and I took a cell biology course, and I just found it really interesting learning about the tools that we have in science, and it left me with so many more questions. And I feel like that's just continually pushed me along of, well, wait, I want to know more about this, and what tools do we have to ask this question, and what tools do we have this? So I find the technology aspect of biology also really interesting, developing these new tools for science. And so after that initial class, that was a big impetus for my research during high school. I wasn't really interested in science at all, to be honest. It was just, took this class, and I didn't really think anything of it at the time until I really got invested in what was really going on cellularly, actually.
So did I see in your background that you've also done a lot of theater and theater stuff?
Yeah.
Tell me what you did.
I have a pretty non-traditional background for a lot of people in grad school. I previously went to a conservatory for the performing arts and studied theater and dance, and worked in professional theater for a little while before my science life. That's kind of another story, but I got a bad injury and had to kind of reevaluate career-wise what I was going to do, and I remembered my interest in science, so I came back to that and continued on with my science career. And even though they seem like very different fields to most people, people ask me, like, how did you transition from doing theater?
I was going to ask the same question. Yeah.
But I think there are a lot of similarities in kind of working with a team, and like the curiosity and openness to do something new, I think, is a lot of what you do as an actor. You're kind of stepping into this new thing and figuring out with your team, like, what can you do with this? And curiosity and innovation is a huge part of science. And I think that's a big part of acting, too. You're building this thing, and in the lab, I'm also building something and trying to figure it out and working with that and working with other people. I think there are some similarities that match to me.
Oh, that's interesting. When you were acting, did you have a favorite role that you performed?
One of my favorite roles was in a musical called Curtains, which is relatively new, so not a lot of people know it, but it's basically a spoof on a musical. And people were so excited in the audience that it made you feel so good to just be like, these people are so happy, and like, here I am dancing. And it was really fun to interact with the audience when, I don't know, everybody was just so excited about it, and I thought that was a really fun connection.
Did you have to sing in the musical? All right. You know?
Oh, no, do you want me to sing, I mean?
Just a few bars, you know?
I don't know about that. I won't be recording any albums anytime soon.
All right, well, we'll put that one aside.
Okay.
All right, congratulations.
Thank you.
All right, welcome to the stage, our third contestant, Kevin Pham from UC Riverside.
For 1,000 years, China colonized Vietnam. For almost 100 years, France colonized Vietnam. For 20 years, the United States fought a war in Vietnam. So today, when most people think of Vietnam, they think of colonialism and war. However, what many people do not know is that this history is just as much about cross-cultural learning. Now, I'm not talking about how pho, Vietnamese beef noodle soup, might actually be inspired by pot au feu, a French beef stew. No, instead, I'm talking about ideas. By the 1920s, a generation of Vietnamese felt humiliated because foreigners had again conquered their country. These 5 Vietnamese thinkers proposed different ways of responding to Western domination. Between them raged a battle of ideas. For example, Phan Châu Trinh argued that the Vietnamese were conquered because they were weak, because they forgot the teachings of Confucius, a Chinese philosopher. He thought that an idea from Europe called democracy could actually enhance Confucian methods of self-improvement in Vietnam. However, Nguyen Anh Ninh argued that the Vietnamese should turn to Indian and German great thinkers as role models in order to strengthen their intellectual culture. They each had different ideas, but they all believed that the way to overcome foreign domination was self-help. You are familiar with self-help books? Well, these guys had a lot to say about self-care, self-improvement, mental health, spiritual health, and even physical health. For them, self-help was a way of strengthening the Vietnamese people to gain political power to become free and independent. Now, what's unusual and brilliant is that they created their theories of self-help by taking Asian ideas like Buddhism, meditation, Confucian family values, and strategically mixing them with Western ideas found in George Washington's biography or French philosophy. Unfortunately, their voices have been forgotten. My project is the first to analyze their writings to show the value of Vietnamese ideas for self-help and politics. They can teach us how to blend ideas from different cultures in order to improve ourselves. Today, in our globalizing world, different cultures are coming into more contact. We need to find out how to pick out the most useful from each, create something new, and use it to improve ourselves, our country, and our world. These Vietnamese thinkers show us how it might be done. Thank you.
All right, so let's start, and let me just ask how you got interested in this particular topic, because you're kind of going in a new direction from traditional history, are you not?
Yes. I guess I would say I am trying to feed 2 birds with 1 scone. So, kill 2 birds with 1 stone. The first bird is that I wanted to rediscover my roots. My grandparents both passed away after I finished my senior year at UC Irvine. And then I decided to move to Vietnam to relearn my native language, Vietnamese, because I was ashamed that I had never really had a conversation with my grandparents. I saw them all the time, but I couldn't really speak to them in Vietnamese, and so I wanted to learn Vietnamese again, and so I wanted to find a project that would allow me to rediscover my roots and to learn more about the history of the country where my parents came from. The second bird is that political theorists have studied a lot of European political philosophy, and recently they've only started to study non-Western political thought, such as China, India, and the Islamic world. It's really those three, but so far no one has written anything on Vietnam and political theory, and so I wanted to contribute to this aspect, and not because Vietnam has been ignored, but because I actually think they have really useful and important things to teach us. So that's why.
So if you were advising President Trump on how to imagine the United States in today's world, what would be your top, let's say, 2 pieces of advice? Put yourself in his head.
The first thing is that if the United States wants to maintain their positive influence on the world, the United States needs to tap into what it has best, which is diversity of cultures. There are many cultures in this country, and we need to take it seriously. We need to reach across the aisle to people of different cultures and learn what aspects of their culture, their religion, their ideas, what they have, how they see the world. And we need to talk and come together and make this country a better place first, and it is because it has fewer blind spots, because we have many different cultures, and I think that's what we can contribute to the world, this model, this philosophy of engaging people across cultures to tackle common problems.
Mm-hmm. And are you, yeah. Are you thinking about how cultures come into contact with each other without also coming into conflict with each other?
And when I did this, when I said contact,
yeah.
Because if we don't make productive, constructive use of this encounter, it might end up in conflict. And so I think these Vietnamese thinkers are valuable because they taught you that you could pick out the most useful from various cultures and synthesize them.
Mm-hmm. And what do you like to do in your spare time when you're not dissecting Vietnamese great thought?
So right now, I've been spending a lot of time with a very close friend, and he has 2 small children, a 3-year-old and a 7-year-old, and they speak French, and so do I. And so I've been reading them a lot of children's books. So I've been in French, and so I'm discovering the world through their eyes and seeing them grow and discover the world. And sometimes when books are not there, I try to tell them stories. Mm-hmm. And so it's really fun.
Yeah, so do you have a favorite children's book?
Yeah, there's one that we read recently called Pluk. It's about a penguin who can't swim and so steals fish from this little Eskimo girl. And the Eskimo girl finds out, and the Eskimo girl hates the penguin, and they get into a conflict. And then the Eskimo...
That's a clash of cultures,
yeah,
right? Yeah.
And then the Eskimo girl's little sister starts sliding off the glacier into the water, and then Pluk comes, and he can't swim because he's never tried, and then he jumps in and he saves her, and he can swim. And then the Eskimo and the penguin are friends.
Well, thank you, and thank you for sharing the story of Pluk. Yeah, thank you very much. All right, now it's time for our 4th contestant. Welcome to the stage, Nyasha Maforo from UCLA.
At 2 years years old, my sister experienced a brain injury that led to 2 weeks in the hospital. But with powerful medical imaging, doctors were able to provide the care that she needed. Now she's happy, healthy, and thriving. In fact, she's graduating high school tomorrow. As I look back at that time, I realized it was a pivotal moment, not only for my family, but for me personally as well. It inspired me to pursue a career in which I could help kids and also learn more about the medical imaging tools that help my sister. So now in graduate school, I'm actively using what I've learned about medical imaging to help kids with Duchenne muscular dystrophy, or DMD. DMD is the most common fatal genetic muscular disorder primarily affecting boys. 300,000 boys are affected worldwide. DMD itself is caused by a DNA mutation that can only be passed down by the mothers to their sons, and this results in progressive muscle weakness. These kids are typically diagnosed before the age of 5 and often require use of a wheelchair by age 12. And because our diaphragm is also muscle, boys with DMD can face respiratory complications. And as this disease progresses, heart problems become the #1 cause of death of these kids. But all hope is not lost. In order to reduce this frightening statistic, this means that we need to look at the heart a little bit closer in order to better understand how this disease affects each boy's heart. And we can do that using magnetic resonance imaging, or MRI. MRI was one of the imaging tests that helped doctors help my sister when she was sick. But in my research, I'm using magnetic resonance imaging because one of its advantages is that we can look at the heart from many different angles, including the cross-section that you see on the right. The dark circular region, also highlighted in orange, that's the muscle that we're interested in. One thing we don't know about DMD is that we don't quite understand when the heart becomes affected. And that's the specific question I'm trying to answer with my research. I'm working to answer this question by analyzing hundreds of MRI images from boys that are healthy, as well as boys with DMD at all ages. I've developed an analysis tool that utilizes color maps in order to look for patterns of muscle damage in the heart. And so far, I've seen that in the muscle region of healthy boys, it's more uniform in color, as you can see. However, in the DMD heart, it presents with an irregular color pattern, indicating where the heart is more affected. These results are really exciting because this tells me that MRI can pinpoint the exact time that the heart becomes affected. So finally, this will impact boys with DMD because doctors all over the world can use this analysis tool to track the disease progression of their patients. And MRI can evaluate existing and new therapies aimed at helping kids with DMD. Thank you.
All right, that's great. So you told us about your family story, but have you always been interested in science?
No, actually I haven't. Very similarly to Angela, I did not get interested in science until very late in the game. I think growing up I had a very limited idea of what a scientist was and what they did because I hadn't really seen it. Also, I come from an African family and it's joked around often that the only careers are an engineer, a doctor, or a nurse, and that's about it. And so I didn't know what that would look like. And so in college I got a summer research internship where I got to study something I was super passionate about, and that was breast cancer, and using magnetic resonance image to better understand that too. And so I realized I was like, wow, this is really cool. So you tell me if I can just, research what I like and be able to share that with my friends and colleagues, every year at conferences, that's really, really exciting. And so that is kind of what got me in, and I was sold on grad school and applied.
And here you are.
And here I am.
Yeah. So if you were talking to a group of high school students about research, how would you explain what it is that you do?
That is a very good question. I think kind of like what I touched on, I would say research is being able to push the boundaries of what we know so far about something you're really passionate about and kind of how that directly impacts the world.
All right, let me go in a somewhat different direction. Let's imagine you're hosting a dinner party.
Oh.
Okay, and you get to invite 3 guests.
Okay.
Who's sitting around your dinner table?
Okay.
Anybody, it can be alive or dead, actually. Yeah.
Okay, that's great. Well, my faith is really important to me, so I'd love to have Jesus at the table. I've got a lot of questions for him. And I would love to have a seat with Michelle Obama. She's a huge mentor of mine, whether she knows it or not. And I'd love to have you there, because I think it'd be great that you propose this, we do it, we talk, and it'd be really cool.
Yeah. Nice touch.
Yeah.
Nice touch. And what do you like to do in your spare time?
While living in the LA area, I've gotten really excited about doing outdoor things. So I've enjoyed hiking, something that my lab loves to do, running on the beach, as well as yoga, cooking, and just experiencing what LA has to offer.
Yeah, so you like to cook?
I do a little bit.
So what are you going to serve at our dinner party? Because I like to eat.
Let's see. That's a very good question. Well, I've been trying to make sushi, so maybe I'll try that.
Oh my gosh.
Yeah.
And what do you think of your what you will do after graduate school? What's your future?
So I'm keeping an open mind. I might go into clinical medical physics, which is what a lot of people in my program tend to do. Maybe stay in academia or industry. So I don't know, I'm keeping an open mind.
Yeah. And so you're at UCLA. Any celebrity sightings that you can?
Oh yeah, there's actually been quite a few. That's one of my favorite things about being in LA. I recently saw. What is his name? One of, right?
Oh yeah, that guy.
Well, it's a retired NBA basketball player, Ron Artest. He was in Whole Foods. Saw him there, so that was really, really cool.
All right, all right. Well, good luck to you. Thank you. All right, let's welcome to the stage contestant number 5, Rachael Harbeitner from UC Santa Cruz.
Every day, you and I are adding more and more carbon dioxide to the atmosphere through our daily activities such as driving. And this carbon dioxide has been accumulating for a really long time, leading it to be one of the major causes of climate change. Luckily, we live on an amazing planet where 3/4 of the surface of our Earth is covered in oceans. And this ocean works to regulate our global climate by acting as a buffer, pulling carbon dioxide out of the atmosphere into the ocean. There are small organisms that live in the surface of the ocean that then use this carbon dioxide to build and grow their bodies. Eventually, these organisms die and their bodies start to sink. And in the ocean, these sinking bodies look a lot like snow. And they accumulate on the seafloor where they settle, they're buried, kind of like a carbon graveyard. But some of these sinking bodies are consumed by these tiny carbon eaters, which you might know of as bacteria. So bacteria can be found everywhere. You and I are more bacteria than we are human. And they're really important for most of our functions. And in the ocean, bacteria, one of their roles is to eat carbon. But we still don't really have a full understanding of what their impact is on how carbon is cycling through the ocean. So to figure this out, we went to sea. We used this really cool giant robot to get to the sea floor. And I actually did experiments feeding carbon bodies to deep sea bacteria to figure out which bacteria are eating this carbon and how long does it take for them to eat it. So we're thinking deep sea bacteria are going to be very slow growers. They live somewhere that's really cold. It's completely dark, and they're living under high pressure. So I thought, maybe in about a year, they'll eat the carbon that I've provided them. It turns out, deep sea bacteria can eat carbon in the course of a week. That's 50 times faster than we had expected. And when these bacteria are eating carbon bodies, it's getting converted back into carbon dioxide. So this carbon dioxide that starts in our atmosphere, gets pulled into the surface ocean, turned into carbon bodies, is sinking, getting converted back into carbon dioxide, and now has the chance to circulate through the ocean, reaching the surface ocean and being reintroduced into the atmosphere. So we know the oceans are working to regulate our climate, and we're still trying to understand what impact do these tiny organisms, like bacteria, have on the carbon cycle, both in our atmosphere and within the ocean. So we can use this information from my experiments to better predict how our oceans are responding to the human impacts of climate change. Thank you.
All right. So, did I see that you also tagged some sharks in underneath the water in Mexico?
So yes, I've been interested in oceanography pretty much forever. And so yeah, I had one of my internships in college. I had the opportunity to tag sharks in the Gulf of Mexico.
Okay, you got to tell me what that's like.
So we go out on a small ship, and we actually set up long lines and baited to catch sharks, and then I actually got to pull them in. We pull them onto the deck of the ship, and we take measurements, figure out age, species, and then stick tags on them to kind of follow where they end up throughout the Gulf of Mexico or beyond.
You stick tags on them? Mm-hmm. So maybe some of the mussel feet would be helpful, right?
The alternative right now is like a piercing of an ear, but you do it to their fin.
You pierce the fin of the shark? Mm-hmm. Okay. Any exciting stories you can relate about that? I mean, how do they react?
Actually, I don't really remember too much. Mostly I remember like fighting sharks to pull them on board.
You said you've been interested in biology a long time. What got you interested?
So I grew up in Maryland, and where I was part of like the Chesapeake Bay watershed. And so the middle school that I went to, we did a lot of outdoor school kind of styled activities. So we were out in the watershed and learning about how, even though we lived maybe a 45-minute drive from the bay, how our life was directly connected to the bay and therefore the rest of the ocean. And that pretty much got me hooked. And I've stayed coastal ever since.
Yeah, is that what brought you to Santa Cruz?
So yeah, so coming to grad school here. So I've done work on the Gulf of Mexico, did work in the Atlantic Ocean. So I figured, why not the Pacific?
Yeah. So if you were the head of the White House Office of Science, and there is one, what would you advise them with respect to climate change and policy about climate change? And small question.
Yeah. So I think one of the main things is that I think it's pretty well established now that climate change is happening. The question now is how much of it is our impact versus what was happening naturally. And I think kind of a lot of what the research that we're doing is trying to say, like, what's the baseline of where we are to understand how are things changing to then determine, do we need to do something about it? Like, which things should we make changes for that we want to try and keep as the baseline, and which things are we kind of okay letting change a little bit?
So any recommendations on that?
Not at the moment.
That's okay. What do you like to do in your spare time besides?
So we're in Santa Cruz, and so this is great access to the ocean, redwoods, mountains. So I spend a lot of time outside when I'm not finishing my dissertation right now. And so I'm also running. I cook a lot. I bake.
Yeah. And what do you see for your future? What do you want to do after you get your doctorate?
So I've applied for a few postdoc fellowships. So hopefully in a month I'll know where I end up next.
Oh, very good, very good. And okay, I'm gonna ask the dinner party question. So you have 3 guests to a dinner party. Who are they gonna be?
I think one of them would be like Sylvia Earle, so having like a female oceanographer kind of early on in their kind of beginnings of being able to do oceanography. This is a really hard question.
Alive or dead?
I think having, that's like as far as I've ever gotten is figuring out I just want to talk to other oceanographers and and how the field has changed and kind of what they were able to do with limited technology. Like whereas now like we have access to like
robots that
I send to like 2.5 miles below the sea floor, and I do things that most people do in a lab or even at the surface, and I can actually use leave experiments and I come back to them like a year later.
That's awesome.
And so it's kind of cool to see where we've been able to progress to.
And it's gonna be cool to see where it goes too. Thank you very much.
Thank you.
All right. Welcome to the stage. Give it up for our sixth contestant, Katie Murphy from UC Davis.
Let's see, by a show of hands, how many of you are looking forward to our lunch today? All right, now keep your hand up if you'd like to be able to eat lunch in 20 years. Okay, that's gonna be 1, 2, let's say just about everybody. My name is Katie Murphy, or as my students call me, the Corn Queen, and I'm working to ensure food for our future. The unfortunate reality is that 20 years from now, we expect 2 billion more people on this planet. That is 2 billion more mouths to feed. Given our current production levels, we simply cannot produce enough food. Lunch in the future could be a luxury. Now, my graduate research at UC Davis is working to solve this problem. How do we grow more food using less resources? We stop losing crops to plant disease. Just like humans, plants get sick, and when they do, our food dies in the field and never makes it to your plate. Now, the nasty, moldy ear of corn that you see up here is caused by a fungal disease. Fungus causes 10% crop loss of corn every year. Now, if we're not having a nice buttery ear of corn for lunch, I can guarantee you that we're eating something that was made with a corn byproduct, or we're eating something that has eaten corn. It is the backbone of the American food supply. What a 10% crop loss looks like is a cornfield that covers the entire state of Florida, rendered totally inedible. If we can rescue some of this crop from disease, we can grow more food. Now, my graduate research has uncovered a new group of chemicals that corn makes in response to this disease. Here, corn is acting as its own doctor, sensing that it's under attack, diagnosing itself with disease, and producing its own medicines, antibiotics really, to fight off this fungus. Interestingly, we've only ever seen these chemicals in corn, not in any other plant. We now know the structure of these chemicals, the genes that control their production, and how they're working as antibiotics to fight off disease. I did so by taking the genes from corn and putting them into a bacteria that I can grow in the lab in order to make these chemicals and figure out what they do. Now that I have this knowledge of these naturally produced plant antibiotics, we can begin to reduce crop loss due to disease. Now, they do call me the Corn Queen, but I love all plants. And so using genetics and breeding, we can make corn varieties and other crops that can make their own medicines. This will reduce our current pesticide use, grow us more food, and generate stronger plants to ensure food for the future. Thank you.
Thank you. Well, thank you for giving us a real earful of...
It was a little corny.
Yeah. So how did you pick corn as your field of study?
Yes, and I normally am outstanding in my field too. It's amazing, I know. So when there you go. That's a slow one. It's a slow one.
We're gonna have a pun down here.
We are, yeah. That's all I have. So don't ask me anymore. So I fell in love with chemistry first when I was in high school. I love chemistry, and when I was an undergraduate, still loved chemistry, but found the lab work to be somewhat tedious, and I couldn't make molecules very well. And so I ended up joining Virginia Walbot's lab. Ginny, thank you so much. And she brought me my love of corn. So she studies corn and how it develops. And I realized that plants are so much better at chemistry than I will ever be in the lab. They can make so much more, and then they do really interesting things with chemicals. And so I then fell in love with corn and got really fortunate when I went to UC Davis that the perfect project happened to be sitting right there, that there was a professor, Philip Zerbe, who wanted to study chemicals in corn and how they help corn fight disease.
Yeah.
So that's where I ended up.
Yeah, that's very good. And what are you thinking about doing postgraduate?
So I want to go do a postdoc, which for those who don't know is kind of what you do after grad school. You go and just do more research. I love research. I love being in the field, in the lab, and asking and answering really interesting questions. But it's also really important to me that my research makes it out to a farmer's field one day. So I hope to stay in research, but I'm also excited to see what I can do with that.
Mm-hmm. You know, it's interesting. One of the things we're trying to do is programs to interest young girls in science and math.
Yeah.
If you're talking to a hypothetical high school class about what it is you do, and you're trying to get the young women to get excited, what do you tell them?
Well, I'd say I think most young women are excited about science and math from an early age. And we see those numbers across high schoolers and undergraduate level. And at the graduate level, we see often 50-50, especially in my program, women and men. And so I often don't think it's a need to excite them. It's a need to just see me and what I've done and to tell them, yes, you can do this. Women are welcome in this field and you can succeed, because I think it's been a lack of having some potential role models, being discouraged to think that even if they're excited about science, girls don't do science. And so I think it's telling them that, yes, you can do it. Here's what I do. I get paid to do research every day. Like, that's the dream.
Yeah.
And so I think that's what I would tell them.
Oh, that's great. And when you're not researching in the lab, whatever, what do you like to do in your spare time?
Well, I like to garden, and I try to plant things that are not corn so I can have a balanced diet. So that's number one. And I just moved to Missouri 2 weeks ago for an internship. So now I'm exploring St. Louis, Missouri, and seeing what it has to offer there.
Yeah, going to go up the arch. And
yes, I haven't gone there yet, but that's my plan.
Yeah, my grandparents are from St. Louis, and my grandfather used to call the arch the largest croquet hoop in the world. So.
Oh, I'll have to go play.
Yeah. And he thought that in East St. Louis, which is right across the river, they ought to construct the largest croquet mallet. So
I'll write a suggestion.
Anyway, so well, good luck to you. Thank you for coming.
Much.
Yeah.
Thank you.
All right. It's time now to give it up for contestant number 7, Ann Deep from UC Merced.
Who here has heard of valley fever before? Raise your hands. Well, for those of you who don't know, valley fever is a disease caused by a soil fungus, once thought to be common just to the Central Valley, but can now be found as far north as Washington, all the way to Mexico. In fact, all counties in California have reported cases. Behind me is a schematic of its life cycle that I've sketched. It starts off in the soil, growing these long threads, and then these threads turn into tiny little subunits that can break off, become aerosolized, and infect you. So anyone who spends time outdoors or works around dirt is vulnerable. Inside your lungs, these tiny little subunits grow, then burst, filling them up with spores, causing your immune cells to migrate, eliciting your immune response. Now, fortunately, only 40% of those who are infected actually develop disease, and it's a very mild disease, kind of like a cold or a flu, and your body can clear it up on its own. But there is a subset of those who develop severe chronic disease, marked by total fungal infiltration and profound exhaustion for the rest of your life. So why is it that some people have very severe disease and others don't? This is an immune mystery. To study this question, I study direct interactions between your immune cells and the valley fever fungus. To do this, I just simply put them together in a dish and I let them interact. Then with a high magnification microscope with a special computer program built inside, I can take pictures of your immune cells to see what's going on. The arrow in white is showing one of your immune cells digesting and breaking down a valley fever fungus. My data currently shows that your immune cells can target and destroy the fungus, but a special unknown activating signal is required to enhance this response. I'm currently investigating what the source of this signal could be. So, why do I do what I do? Well, the reality is, if you traveled up and down California, you've probably been exposed to valley fever at one point in your life. What's worse, if you go to the doctor right now and you have a positive diagnosis, that doctor can't tell you if you can clear your infection or you'll become chronic. My research addresses this gap in knowledge. If we can figure out what is boosting our immune system, the source of that activating signal, we can enhance our immune response, paving the way for better drug development and better therapeutics. What's worse, chronic valley fever today is still treated with the same drugs from the 1980s, and patients have described these drugs as akin to setting their nerves on fire. So we need to solve this problem. And on a more personal level, I study valley fever because as a Vietnamese immigrant and a transplant in California, I've seen what this disease can do to communities without intervention. And so to honor the people who've built our homes and put food on our table, I study this disease today. So, to clear or not to clear? That certainly is the question. But soon, we can say we have an answer. Thank you.
All right. I spent a lot of my adult life in Arizona, and valley fever is a big problem there.
It
so
is,
yeah. What got you interested in science at all? Were you always interested? Did something turn you on? What happened?
I have kind of like a non-traditional background. I started my sophomore year actually doing linguistics research in political metaphor. And so I worked with this great professor, Teeny Matlock, and part of my project also kind of like segued off the side to look at climate change communication actually, which kind of sparked my interest in science communication overall, but also science communication to the Central Valley. So when I was looking for graduate school programs, I thought to myself that I really like language, but I also love community engagement and civics, and I love science. So what's a project that will let me do all of that? And Valley Fever is perfectly that. It gives me the science component so I can ask these great questions, but I can also go out into the community and talk to the citizens of the Central Valley as well and share with them the knowledge that's like right there in their backyard, literally. Yeah.
And when you finish your graduate school, what's in your future? What are you thinking?
So I love teaching, so if I could stay in academia, that would be great, but I'm also super open-minded, and kind of like a quiet passion that's starting to come out is that I would love to work in the NIH or the CDC and work in science communication, because I feel like all this great work that we do is like super awesome, but I know people from the Central Valley, who don't know that Merced is doing Valley Fever research. So if we can communicate that would be like super great.
Yeah. And in your spare time, when you're not researching the cure for Valley Fever, which would be awesome, what do you like to do?
A lot. I love to read, I love to write, but I also really like to play video games. Yeah.
Which one? You got a favorite?
I do. Overwatch. So, thank you, my people.
And you like to read. What are you reading now?
Mostly, I read a lot of fantasy. I'm catching up again on Game of Thrones. I'm rereading it just because I'm up to date on the TV show, but the book always has more details,
So are you watching this last season?
Yes, I am.
Yeah, what do you think? What's gonna happen?
I don't know. Every time I watch it, I just end up a crying mess. So really, I'm just taking it as it comes, honestly.
Yeah. 2 more episodes.
Yeah.
So, do you ever binge watch?
Yes.
Yeah.
My professor doesn't know this, but during those long incubation hours, I will literally just sit there and just eat through 3-4 episodes of any shows. Yeah.
So you like a long incubation period?
Honestly, I make the most of it. How about that? I make the most of it, yeah.
And let me ask the dinner party question.
You know what? I'm ready. I'm ready for it.
Because you kind of knew it was going to come. Yeah.
Well, first, Anthony Bourdain. Honestly, rest in peace. I really wanted to meet him. He's kind of like my hero because he combines the world of art, food, culture, and politics so well. And I'd love to just have conversations with him. I would love to meet.
You'd probably get a pretty good meal too.
I would be cooking, so that's even more pressure.
Pressure's on, yeah.
Yeah. Probably Oscar Wilde, the poet, just because I love his poetry. I love the writing that he's done. And if records are true, he's kind of a party animal, so that might be interesting,
Yeah, he and Bourdain would really, I think.
Oh, yeah.
Yeah.
Oh, yeah.
Yeah. Can imagine, I'm imagining that. Yeah.
And unfortunately, to throw him into the situation, Jeff Kaplan from the Overwatch team, he heads the entire development and direction of the video game. And so it would be kind of two of these great, robust thinkers and talkers and these two nerds in the corner. Yeah. So.
Okay, well, that sounds like it would be interesting. All right.
It would be, yeah.
Thank you very much.
Thank you so much.
All right. Please welcome to the stage our 8th contestant, Nancy Freitas from UC Berkeley.
What if I told you that bacteria, tiny microbes we can't even see with the naked eye, are partly responsible for some of the biggest climate changes we are seeing on the planet? Bacteria are so tiny, I could hold 1 billion of them in a handful of soil. Think about the number of handfuls of soil there are in the Arctic, where I work. All of that bacteria is changing our climate. On the left, you can see the top layer of permafrost in the Arctic. It's made of plant material, frozen for thousands of years. But the next picture shows that climate change is thawing this permafrost. Bacteria that were dormant are waking up, and they are eating plant material. When they do this, they produce carbon dioxide and methane, potent greenhouse gases. This graphic shows northern Alaska, where I work. And the brown is land and the blue represents lakes. Now, scientists used to think that the soils deep under these lakes were unaffected by climate change, but we're learning that they were wrong. And this is a huge problem, because as you can see, lakes cover 40% of the area of the Arctic, but we don't know how much greenhouse gas these deep soils are releasing. So the climate models that we rely upon could be vastly underreporting greenhouse gas release in the Arctic. The goal of my research is to collect this missing data. On the left, you can see part of a 60-foot-long sediment core taken from below an Arctic lake. The vials are samples taken at various depths. On the right, I put these samples in jars. I expose them to different temperatures. Using the syringe, I can capture and analyze the carbon dioxide and methane that the microbes in the sediment are releasing. These data will be fed into a large climate model. It's being compiled by 4 national laboratories. And it is crucial that we make these models as accurate as possible. It is the only way to understand what is happening with climate change. And the solutions are there. We even know what some of them are. It's my hope that my work will motivate humans, which are much larger and much smarter than microbes, to put our solutions into action. Thank you.
All right, now, did I see somewhere that you grew up in Arizona?
I did grow up in Arizona.
Yeah, yeah, and you still have family there by any chance?
I do, yeah.
And was your family there in the late 90s, early 2000s?
Yes, they were, and we've met before.
Awesome. Yeah. I was gonna ask, did they vote for me? But that's an inappropriate question.
The answer is yes.
Yeah. So how do you get from Arizona to the Arctic and Alaska? What was the journey there?
So I actually, similar to other people, I wasn't interested in science very much. I liked math, but I didn't have a lot of female role models in the sciences. And I had a teacher in high school who was incredible. And she pulled me into research and got me established in a lab pretty early on. And through some of my lab work in undergraduate, I got sent to the Arctic. And I started doing work on lakes. And it's kind of taken off since then.
Yeah, yeah. And so how did you make the jump from doing research up there to climate change? So tell us about that journey.
Yeah, so the Arctic is kind of on the frontier of climate change. We know that climate change is happening, and the Arctic is warming at twice the rate that anywhere else in the world is. And so these changes are happening very, very quickly. And like I was saying, it's crucial that we study them and that we actually start doing something about this.
Yeah, yeah. And are the models capable of analyzing not just the change that has occurred, but the rate of change going forward?
So these are projections, right? And we are looking at current trends in the environment, and we're trying to understand how they might change moving forward. And the IPCC which releases climate reports every few years, has different scenarios for warming that are possible. And so using those different scenarios, we can come up with potential effects that we might see.
Yeah, yeah. So pretend for a moment that I'm President Trump.
Okay, no.
Okay. You're advising me on science policy affecting climate change. What are you going to tell me?
I'm going to encourage you to go to some of the top universities in the country and to listen to what scientists see every single day out in the field. I don't think it's effective to try to tell people that climate change is real when they don't necessarily see that it's real. But if people are able to experience what scientists experience on a daily basis, it would be pretty hard to deny.
Yeah.
And I think policy will follow after that.
Yeah, yeah. So, yeah. And you'd bring them to Berkeley for sure.
Oh, of course.
Yeah.
And Arizona, the University of Arizona. gotta do both.
Yeah, very good. What do you like to do when you're not exploring the changing climate?
I play ultimate frisbee, which is a very up-and-coming sport.
Yeah, what I see that phrase, ultimate frisbee. What is it?
Ooh, great question. I love this question. This is much better than the dinner party.
which may still be coming at you.
Please don't ask me that question.
Yeah.
So ultimate frisbee is played on like a soccer-sized field, and it has 2 end zones, just like football. And there's a kickoff, kind of, where one team throws the disc to the other team, and then they start playing. And it looks kind of like rugby when they're playing without the contact. And so you try to score in an end zone, and you play to a certain number of points.
How many people on a team?
Teams usually have between like 20 and 25 people, but on the field you have 7 for each team.
Okay.
Yeah.
So you have 14 people going after the disc.
You do.
Yeah.
Yeah.
All right, so, all right, you're gonna get it.
No. So my time yet?
Your time technically is up, but I gotta ask. 3 people you're inviting to dinner. Who are you gonna have around the table?
I have a question. Can I squish people can I make multiple people one person? It's a theoretical dinner party, right?
Be as creative as you want to be.
Okay, great. All 4 of my grandparents would be in one person, because I would really like to talk to them again.
Yeah.
And I would like past President Obama and Michelle in one.
You're doing well.
Yeah. I just got to cover all of my bases.
Yeah.
And then I didn't have an answer for the third one. Probably my parents.
Yeah. Oh, squished into one.
Oh, yeah. I'm squishing people. They can share chairs,
Yeah. It's going to be a crowded dinner table. Okay.
Yeah.
All right. Well, thank you very much.
Yeah, thank you.
Okay. All right. Let's give it up for contestant number 9, David Wu from UCSF. David.
Anyone can get a concussion. You don't have to play for UCSF football. Every year, over 1 million Americans go to the ER for just falling and hitting their head. And even one concussion can cause long-term problems. Imagine, bam, you just hit your head. At the ER, you ask a set of questions. What's your name? Where do you think we are? Can you tell me today's date? Right now, a questionnaire is our best diagnostic for concussion. A CAT scan of your brain is not sensitive enough, so you're often sent home, unsure if you had a brain injury. What if we did have a sensitive test? Bam, you just hit your head. At the ER, you take a blood test, and it tells you, do you need medical care, or can you just go home? 20 years ago, a simple blood test revolutionized how we catch heart attacks. A unique molecule from your heart, troponin, was caught leaking into the blood after a heart attack. If that showed up on a blood test, doctors know, hey, this is serious. When you hit your head, your brain also leaks molecules into the blood. And if that showed up on a blood test, I think doctors would know, hey, this is serious. Sounds good, right? Why haven't we done this? Well, for a heart attack, it took decades of guesswork just to find troponin. I got to graduate. So I I devised a systematic two-part strategy to find the troponin of concussion. Secret weapon 1. Instead of looking for the usual suspects, we went unbiased. We came up with a way to profile thousands of molecules at once, and we looked in the blood of 100 people who just had a concussion, 100 people who didn't. Secret weapon 2. Under your skull, your brain is not naked. It has a soft covering, the meninges, that often gets overlooked because it seems really boring. We think it's the perfect sensor for mechanical head injury. It's thin, delicate, and while the brain is sealed from the blood by a barrier, the meninges is outside the barrier. If injured, it could leak right into circulation. So we analyzed the human meninges for the first time at high resolution. We charted a map of all the molecules made in this special covering. This uncovered new molecules found nowhere else in the body. And here's what surprises me the most. This actually worked. Those new molecules from the meninges, we caught them spilling into the blood after concussion. Even if your CAT scan couldn't catch it, our molecules were there, telling us, hey, this is serious. Look, we still have a lot of work to do, but we hope this brought us one step closer to the blood test we sorely need, something everyone can take if they hit their head. No one should fall through the cracks because anyone can get a concussion. Thank you.
All right. Okay. So, David, what got you interested in science to begin with?
Yeah, so it's a long story. I actually did not like freshman biology of high school, so I was planning to do something totally different.
What were you planning to do?
I was going to take like chemistry or something, but the girl I had a crush on was going to take AP Bio. So then I took AP Bio, and that led, I actually didn't even end up in the same class. She dropped the class. It was a mess. But that started the chain of events that got me right here. So here we are.
Yeah. So you should be grateful to her.
Oh, yeah. Yeah, absolutely.
Yeah.
If you're out there, thank you.
Yeah.
Yeah, thank you very much.
And if you have a high school class in front of you and you want to explain or excite them about doing science, give me the pitch for science.
Yeah, I think science, most people don't realize you get to choose a problem that you care about a lot, that you're excited about, that you want to solve, and then people give you money and you go solve it. Mm-hmm. It's like, it's the dream job, right?
Yeah.
It's like, it's not people, there's no textbooks that you're reading, you're writing the textbook.
Yeah.
And I think I didn't realize that until very late in my studies, but it's really incredible. Like we're creating the knowledge.
Yeah.
And there's no other job like that.
Yeah. And when you're not doing science and researching concussion, how do you like to spend your spare time?
Yeah, so I get randomly inspired every few months. Currently, I've been making craft cocktails. So, yeah, my roommate's actually here. He's heroically volunteered for a lot of these experiments at home because I don't actually drink very much. So he kind of helps me out with that.
I'll volunteer.
Yeah. Great. What kind of drinks do you like? Like sweet, sour, sparkling, strong.
Sparkling, dry.
Okay.
That kind of stuff.
Yeah.
Got some ideas?
Yeah, I got one for you. So St. George's Distillery in Alameda, they make this incredible liqueur called Bruto Americano. You add that over either some sparkling wine or like ginger beer, throw in a lime. It's incredible.
Oh, that's great.
You'll love it.
That's great.
Yeah.
I'm so glad I came today. I got a cocktail recipe. Yeah. So, do you have brothers and sisters?
It's just me. So we grew up in New York City, and New York City has a one-child policy. Just economically, all my friends growing up, we were all like, no one had brothers or sisters. Then I moved to the DC area, and like everyone had a brother or sister. I was like, what's going on?
Yeah.
So we actually share some commonalities. We both went to UVA.
Yep.
Not at the same time. And before coming to UC, we both worked for the federal government in DC for a little bit.
Yeah.
So,
What did you do for the feds?
So I was at NIH. So in 2013, there was a government shutdown, and I am pleased to say that I was considered an essential employee at the NIH during that time.
So you got to continue to work, but weren't getting paid while you were working.
Exactly.
Yeah.
It's quite an honor.
Yeah. You know, that's how those shutdowns work. If you're essential, you got to keep working, but you still don't get paid.
Exactly.
So, yeah. If you're not essential, you don't have to work, and then you get paid too. So, you know, it makes one wonder about essentiality.
It does.
If that's a word. All right. Well, thank you very much.
Thank you so much.
All right, great. We're up to our 10th and final contestant. Give it up for Rachael Rosenzweig from UC Irvine.
When you hear the words swarming with deadly bacteria, you probably imagine environments like murky lakes with warning signs, or rat-infested landfills, or basically anything in a public restroom. But what if I told you these same deadly bacteria also infest surfaces and environments we rely on for our own health and safety, such as hospitals? Every year, there's 2 million cases of hospital-acquired infections due to contaminated medical devices and even implants. This leads to 100,000 deaths and over $20 billion in healthcare costs in the US alone. Our only current solutions are chemicals and antimicrobial drugs, but they're being overused and overprescribed and helping these infectious microbes like bacteria and even fungi develop a rise in antimicrobial resistance. The World Health Organization has declared these infectious microbes as a dominant threat to global health and called them superbugs, with death rates predicted to surpass cancer in 30 years. So how do we actually prevent these death rates and defeat superbugs? My research aims to engineer surfaces for commonly contaminated medical devices and implants that are antimicrobial without chemicals or antimicrobial drugs, or what I like to call superbug kryptonite. To do this, I go to who seems to have all the answers: nature. The wings of insects like butterflies, dragonflies, and even cicadas may be beautiful to look at with our eyes, but what we don't see is the superbug kryptonite. When you zoom in their wings, they have an array of tiny, tiny nanopillars, so tiny they're 1,000 times thinner than your own hair. You can't see or even feel them. But infectious superbugs can. When they land on these wings, they can't grow, and they often rupture and die. To translate these nanospike surfaces onto commonly contaminated medical devices like contact lenses, I use a low-cost technique called nanoimprint lithography that just heats up a material surface and applies a stamp with nanospike patterns on it, like using a stamp to seal wax on a letter. So when I test these superbugs on these commonly contaminated medical devices, where the only thing that's changed is the physical surface texture, they also can't grow, and they rupture and die. So if we can apply these antimicrobial nanospikes onto commonly contaminated medical devices and even implants against antimicrobial resistant superbugs, we can apply this tiny solution to this huge deadly threat. Thank you.
All right. So was it hard going 10th?
Yeah. It was fun. I got to see all the talks and I got to learn stuff.
Yeah. And you got a kind of a preview of some of the questions, but so I'm going to throw a new question at you. You talked about superbugs and kryptonite. If you were a superhero, what superpower would you want to have?
As a graduate student, I would love to have the superpower of speed reading. Because then I could read so many papers, etc. But because But realistically, for fun, I'd probably want to teleport so I don't have to order a lift or or drive.
You can avoid the traffic.
Exactly, yeah.
Yeah, I'm with you. When you're not doing research, what do you like to do?
So we're at UC Irvine right now, which is in Southern California. One of the most things is there's always these new and exciting workouts to do, which kind of gets the endorphins flowing, so it helps you de-stress. So I've recently got into aerial acrobatics as well as like infrared heated yoga with weights. So it's fun to try different things.
Okay. Aerial acrobatics. Okay, what does that entail?
Aerial acrobatics is basically like, it's also called aerial silk. So you have these two silks from the ceiling, you hold on to them, and you feel kind of like a kid at the jungle gym again. That's why it's also very fun, and you just lift yourself up, do different poses and lifts, and have a workout at the same time.
Yeah, can you do that in a room with the infrared heat so that you get it both?
It'll get kind of sweaty, so I might slip. Probably not.
So here's another one. So tell us something about yourself that your graduate advisor doesn't know.
So I've had this question before. I've thought of it. I think my graduate advisor probably knows me better than I know myself. But one thing that a lot of people find amusing is that I guess I'm a very serious minimalist. For instance, I only own one piece of furniture besides my mattress. I only have one app on my phone. One of everything.
And what has caused this syndrome to happen?
Just my brain sometimes feels cluttered, so when there's less physical clutter around, I feel so much calmer.
Yeah.
It's easy, just clean and feel good. So
yeah, so you got to tell me, what's the one piece of furniture?
My dresser.
Okay, okay. And where do you see yourself going after you get your degree?
So I'm kind of nearing the last stage of my PhD. So hopefully after this, I'm hopefully to do a postdoc, because now that we know kind of what's happening, the effect of these surfaces, the next step I want to look into is how it's actually happening. What is the actual mechanism? So it'll be great to do a postdoc to kind of continue that work that we've started. And then eventually go into academia and be able to actually write grants and all these new ideas we have and have a team of other grad students who can do the work.
And have you always been interested in science?
Not necessarily. I, like many of the contestants, kind of grew up not really knowing any scientists or engineers. So not totally, but then of course I fell in love with science and engineering as I went through college, and I couldn't really imagine myself in any other field.
Yeah, did you have a particular class or instructor that turned you on, or?
I always liked science and engineering, like most of the graduate students who go into there, but it was probably just a regular no-grade credit seminar that I took as an undergrad for intro to nanotechnology in the material science field, because I like nanotechnology because it's
minimalist.
Yeah. Oh yeah. Because you can't see it; it's invisible, but you're still solving a problem. And I didn't think of that. That is probably why I like it.
Yeah. and okay, you're gonna get the dinner party question, because who's gonna sit in the one chair that you have at your dinner party? So who's your guest at your dinner party?
Actually, I don't even own a chair or a desk. But we can, if there were chairs and real furniture. So I've been asked this question before, and the first person would be Michelle Obama because of her experience, and to increase diversity, the second guest would be George R.R. Martin, because just to kind of ask him some questions on what he thinks what's going on in Game of Thrones right now, and maybe what
maybe he understands what's going on.
Yeah. And then the third person, because I'll feel kind of starstruck and awkward, so I want Stephen Colbert to be there to facilitate the discussions.
That would be awesome. All right, well, thank you very much.
Thank you.
Let's give it up for all our contestants. Great job, everybody. Great job. I'd like now to turn the podium over to UC Provost Michael Brown.
Thank you, President Napolitano. And thanks to our wonderful competitors, our wonderful graduate students. Let's give it up for them again. I mean, that was just simply exceptional. And I have to say at the beginning, even though it's not in my notes, thanks to our wonderful judges, I would not want to be you. This is difficult, I think. I'm so impressed with the quality of presentations. Now to the undefiled voting and the outcomes on these awards. I want to thank again Thermo Fisher Scientific for sponsoring the cash awards today. This is new and wonderful, so thank you. We will announce the People's Choice Award winner, the third place winner, the second place winner, and the grand prize winner. But I want to say they're all winners. They're all winners. And by the way, the grand prize is called the Slammy. Okay. And I want to thank everyone in this room, those of you who are online, who have took the time to vote. And we're now going to announce the People's Choice Award. And I have it right here. The award goes to... La La Land. Thank you, Regent Perez. Very good. The name is Ann Deep. Ann Deep from UC Merced, and Dean Marjorie Zatz. Pictures, please. All right. Yes.
What do you announce?
This is the third place winner. Rachael Harbiter from UC Santa Cruz. Dean Lori Kletzer, the grad dean. That is awesome. Now, the second place award goes to David Wu, UC San Francisco, Liz Watkins, grad dean. And now the first place winner and the Slammy goes to Katie Murphy, UC Davis, Dean J.P. DePlank, grad dean. And we have one other award, and this we award as a consequence of the student winning. We award to the graduate dean associated with the campus that student comes from, J.P. DePlank, the Perpetual Plaque Award. You are memorialized. Wow, that was just too much. So Now I have to find out where I am in my notes. Okay, then I'm almost at the end here. On behalf of President Napolitano and all of us at the Office of the President, we want to thank again our wonderful event partner, LinkedIn, for hosting the great. That's right. That hosting this Grand Slam event at their wonderful, beautiful. If you're not here, those of you online, you're missing something. It's a beautiful San Francisco location, and for working with us again to present this Grad Slam award. We just couldn't ask for better partners. We appreciate the work done by so many on behalf of this event. There are far too many to identify by name, but there are some that I want to. Certainly the whole LinkedIn team. There's more than probably more than Farhan Saeed, but I do want to acknowledge him. Where are you at, Farhan? Okay. Katie Farrick, you're here. That's right. She is Senior Director of Workplace, Community, and Environment. Let me turn my notes and make sure I got the whole name. So we thank you for your leadership and your commitment to partnering with us, Katie. They have been with us for a few years, even when they were still constructing this building. And there's an audiovisual team associated. Where okay, we have to thank them. Much love. And I want to give a great big thank you to our folks at the University Office of the President for making this event happen. There are many hands involved in this, and not just hands. Heads, because there's a lot of thinking and planning associated with this, but more than that, their hearts, their commitment to this event. So I want to acknowledge them, particularly the marketing and communications team and our graduate studies dream team. And I want to name Sandra Wolf. Don't you be hiding. That's right. Lisette Lim. Yes. Yuri Cesar Kaptovich. Did I get it? Kaptovich? Okay. And Vanessa Moruya. So let's give a collective hand. They worked hard to make this happen this year. And finally, I want to thank each of you because you actually bring an energy and a spirit to this event and even laughing at our bad puns. That's all right. So we want to thank you for your support, your energy, and hopefully for those of you who didn't know and understand graduate education and its importance, you have a greater sense of it now. And for those of you who already did, you will be ambassadors of that out in the world at large, because most people maybe don't know. And you're here to be able to translate that for them. And what graduate education at the University of California means. All right. So we hope that you will take that energy and insight and find ways to support graduate education in your own space and in your own way. So with that, we're going to see you next year, right? Right? All right. Thank you very much. And thanks again to our judges.